All right, then I think I will take it away for diversity and stability team. So our team is going to present our progress on the research question about the role of diversity in oh, the role of diversity in ecological stability across trophic levels and ecosystems. Next, please. I'm going to introduce our really amazing team who work with different taxas and different ecosystems. So, from left to right, we have James, Noam, Katherine, myself, Pooja in the center, and then Kelsey, Juliana, and Junna Wang, people at different stages of their PhD or a postdoc in there. And I would really like to thank our mentors, Laura D. and Forrest Isbell, who have been helping us out to think through this process of relating this diversity and stability questions. Next, please. So I don't think it should come as a surprise to many of you in the crowd that we are losing biodiversity at an alarming rate. But what does it mean? Like, what does it mean for an ecosystem level? If we are losing a lot of biodiversity, how is it going to affect the stability of our ecosystem or functioning of our ecosystem? When we say stability, what I mean is how much variability do we have in some metric of ecosystem across a baseline? So historically, people thought that the more diverse an ecosystem is, the more stable it would be. But then Robert May came with his mathematical models and showed that that's not the case. Through his models, he showed that it's not always the case that complex systems are more stable. So that led into a plethora of research that went into understanding what are the drivers of these diversity stability relationship and what are the mechanisms that are working underneath that hood of just talking about diversity. And some of the mechanisms that have came up are like buffering effect, that different species in a community can actually buffer for the effects of environmental change. So some species can perform good under one condition, another species would perform under other conditions. So eventually we end up with having more stable ecosystems. But and that's how diversity can actually lead to stable ecosystems. But we can also have another way in which diversity can affect ecosystems. Stability is the performance enhancing effect, which says that under certain conditions, actually one species is able to perform better because those conditions favor performance of that species. So we might have more variability across environmental changes. So, most of these mechanisms and patterns of diversity stability have been studied in grassland ecosystems or just on one trophic level. And when I say trophic level, I just meant by the producer level. So that's where most of the research have been done in past. And when we came together as a group, we thought like, why don't we test these things across multiple trophic levels, multiple ecosystems, and look at stability in different ways. Next, please. So when we were started thinking about stability and what are the metric that is going to represent what stable ecosystems actually mean, so we brainstormed on that and we came up with two of the metrics. So the first one is what traditionally it has been used in diversity stability studies, which is called aggregate stability metrics, where basically you take the whole community and have some kind of metric that represents total value of that metric for the whole community. For example, it can be just total biomass or total number of individuals, and you just track that metric over time and see how variable it is across time. So that has been what traditionally it have been used in most of these studies. But when we were thinking about these questions, we thought of what about the composition of the community? You have different species that come in and out throughout the time, as well as the relative abundance kind of changes quite a lot. So we came up with a metric of compositional stability, which looks at community in a multivariate space and tracks the composition and the relative abundance of community over time. And that led us to ask different questions, which Juliana is going to cover. So, passing the baton to Juliana.
So, as a group, we wanted to sort of build on these ideas that Pooja outlined so nicely to ask three general questions using synthesized long-term data. And the first question was thinking about how these diversity stability relationships influence multiple trophic levels and whether the diversity of one trophic level could affect the stability of another, specifically thinking about whether producer diversity can affect consumer stability and vice versa. Our second question, we were interested in how these relationships varied on whether we define stability using aggregate measures, so that sort of fluctuation in like total biomass, versus compositional measures, thinking about community composition shifts, shifts in relative abundance, and species turnover. And then our last question is sort of thinking whether these relationships are different in aquatic versus terrestrial systems, given that different sort of primary forces and drivers are at play in both of these systems.
Okay, yeah. So to answer these questions, we looked for pairs of producer and consumer data sets. We focused on finding long-term time series data that recorded community data for primary producers and their consumers. So an example of this would be a data set recording grassland plant community composition, and then a corresponding data set recording the insect community composition. And so we aim for these pairs of producer and consumer data sets to be spatially and temporally paired. So for example, plant and insect communities sampled from the same plots during the same years. And then we've aimed to find pairs of data sets within an LTER that overlap for in monitoring for at least 10 years. And so using these criteria, we found 8 pairs of data sets, which you can see on this map here. 6 of these are LTER sites, and we've supplemented with 2 non-LTER sites. So we have 3 terrestrial sites, KBS, Kanza, and Cedar Creek. And then we have 5 aquatic sites, Moorea, Santa Barbara, and Georgia coastal LTERs. And then our 2 non-LTER sites are the U.S. Virgin Island Coral Reef Monitoring and Australian Institute of Marine Science Coral Reef Monitoring. And so our first major effort has been to harmonize these data sets, cleaning up species names, making sure data sets are spatially and temporally paired, and then dividing them into these broad aquatic and terrestrial categories. And so this is just a quick example of what this looks like. So each row represents the abundance of a species at a site for a given year. And we've added a couple of helpful columns to facilitate easy filtering, such as what feeding group the species is, what habitat type it's sampled from, and then what units of abundance it's measured in. And so we're using this harmonized data set to calculate our stability metrics to answer our questions. Right now, we're using all of our data sets except for the Georgia coastal LTER data, which we're still in the process of working on. But after we're done, we're going to publish this data set to EDI for others to use as well, because this is a really great long-term data set recording paired producer and consumer communities.
Great. And now, thanks, Katherine. I'm going to talk about our workflow and a little bit more on the math behind our stability metrics. So the first thing that we did was harmonize those data sets, and then we were able to calculate a diversity metric. And right now we're just using species richness, but we intend to expand that to other diversity metrics. Then we also calculated aggregate stability and compositional stability, doing the 2 different methods that I'll describe next. And then we used structural equation models to get at those pairwise comparisons that we've shown you in the last couple of diagrams. And then finally, we're going to be interested in comparing aggregate and compositional stability, but also comparing these trends across systems. All right, so for our aggregate stability, as Pooja kind of laid the groundwork for really well, we use a traditional metric called coefficient of variance. We use the inverse of it, where the standard deviation of some abundance metric like total biomass or total counts is divided by the mean of that data set over time. And so that gives you a simple single metric to discuss stability in terms of system level. For compositional stability, we use the EcoTraj package, which is being pioneered by Miguel de Casareas in Spain and his team. And as said, this is using multivariate data and using species assemblage matrices that will maintain the multivariate complexity of a biological community, but then reduce it down to a simple single metric. So what we used were these interannual vector lengths. So these plots are very similar to NMDS plots, where they do Bray-Curtis pairwise dissimilarity, but then we drew vectors between the consecutive years and measured that distance as a metric of change or stability. And so I'm now going to pass it off to Junna, and she's going to dive into some of our results.
Yes, thanks to our team on data harmonization, also our team on data analysis and writing very cool functions to get very exciting results here. So here I have an example about diversity stability relationships from each study site. So in this figure, you can see is the values of species richness vary a lot across those LTER sites. The same variation also existed in the stability metrics. So in order to make those diversity and stability metrics comparable with each other, so we do this standardization. After this standardization, you can see that the figure in the last slide after standardization is in this slide. So the diversity and stability matrix is very comparable across these sites. For some metrics we used, we found that the slope between the richness and the stability in different study sets, they are so similar. This indicates that some consistent pattern arise from all of those sites. With this standardized data, we build a component model for our structural equation modeling. Next slide, please. This is the component model of our structural equation models. So we assume that our stability matrix is a function of producer richness, consumer richness. We also add the random effect of study size to the slope of producer richness and to the slope of consumer richness. So this is how do we are going to present our results. Before that, I want to indicate how to read the results from the structural equation models. So in this figure, this double arrow is a double-headed arrow. It means correlation. So single-head arrow indicate the direct effect. The two vertical arrow it indicate the direct effect between diversity and stability within the producer community and within the consumer community. So the two cross the arrow indicate the diversity stability relationship across producer and consumer community. Next. Oh, sorry. The lines, the dashed lines indicate the insignificant effect. Solid lines indicate the significant effect. And the blue color indicate the negative relationship. Sorry. Blue color indicate the positive relationship and red color indicate the negative relationship. Okay, first we see our results about the diversity aggregated stability. So for the terrestrial ecosystem, so we look at the 4 single-head arrows, which indicate the direct effect of diversity on stability. We have noticed that for terrestrial ecosystem, all the 4 arrows are dashed lines, which means the relationship is insignificant for the terrestrial ecosystem, both within the producer community, within the consumer community, and across the producer and the consumer community. However, in aquatic ecosystem, we had very different results. So first is the diversity stability relationship within one group, within the producer community and within consumer community. They are significant, which is indicated by the solid lines, and they are positive, which is indicated in the blue color, which is different from the terrestrial ecosystem. However, we do see 3 similarities arise between terrestrial ecosystem and aquatic ecosystem. First is the cross producer and consumer community, this cross-group diversity stability relationship, all insignificant and weak. Second is the correlation between producer richness and consumer richness. They are all positive and significant, indicate that the richness of the one community may track the richness of another community. However, the correlation between producer stability and the consumer stability are both insignificant. So next, Noam is going to present the results about the diversity and the compositional stability.
Yeah, so I'm going to move a bit into the compositional stability side of things. So remember, Junna was just referring to aggregate stability, which is that more standard metric of looking at the stability of total biomass or abundance. Whereas compositional, we're tracking things in the multivariate space. And so the first thing that we wanted to look at before we even started modeling was whether these two metrics are correlated with each other. And that's what you're looking at here. On the left is going to be your producer stability metrics, and on the right is your consumer stability metrics. And we looked at this within our various sites as well as if you combine them. And generally speaking, what we found is that kind of contrary to our expectations, our aggregate stability and our compositional stability metrics had fairly strong positive correlations with each other. And so that was actually, like I said, surprising and quite exciting. And so then we started thinking about it in terms of these broader multi-trophic relationships. And so I'm going to start first by focusing on our terrestrial systems, just because that's been more commonly studied. And this is what we saw with our aggregate stability metrics, right? Was that there was that positive correlation between producer and consumer richness, like Junna said, but nothing else really showed a strong or consistent relationship. And then when we switched that to focus on our compositional stability metric, we find that these trends are actually quite similar. There are some flips in the directionality of those relationships, but again, we're still looking at a bunch of dashed arrows, not statistically significant in that sense. However, when we move to our aggregate stability for aquatic systems, if you recall, we saw strong, significant positive effects within consumer and producer groups of richness on stability. But when we look at compositional stability, we find that these are actually gone. So there's no longer a strong predictor of producer richness on compositional producer stability, nor is there a strong relationship between consumer richness on compositional consumer stability. However, interestingly, we do see that producer stability and consumer stability are tracking each other when we use this compositional stability metric. And it's possible that's because our aquatic data sets are actually quite complex in terms of their species matrices. And so they're sort of tracking that high richness and high diversity relationship more closely in terms of the stability multivariate space. And so I'm going to pass it off to Kelsey to kind of wrap us up and take us home.
All right. Hi, everyone. I'm going to take us through our preliminary conclusions and what we see as our next steps as a group. First, to address that first question, we found that producer and consumer richness are consistently correlated, but that producer and consumer stability are rarely correlated. To address that second question, we found, surprisingly and excitingly, that the aggregate and compositional stability are linearly and positively correlated. But we found a lot of variation in the strength and direction of diversity and stability relationships. In terms of those ecosystems, we found that aquatic systems tend to show stronger relationships than the terrestrial systems. We also found in terms of metrics that there was weaker within group diversity stability relationships for compositional stability in the aquatic systems. Finally, so far we have not detected strong relationships between producer diversity and consumer stability or vice versa. Next step. So in terms of data harmonization, right now we only have 3 terrestrial data sets that we're working with. So we'd really like to find some additional ones to strengthen the power of our statistical analyses. We're going to be looking at NEON data sets, but if anyone here has some ideas of some additional data sets that can meet our criteria, please let us know. Today we presented on preliminary results, but we have a few ideas for other steps we want to take in our analyses. The first, as James mentioned earlier, are looking at alternative diversity metrics. Right now, we're looking at species richness, but we're interested in exploring Shannon diversity and effective richness. Next, we want to explore calculating compositional stability based on Jacardian dissimilarity. Right now, we're using Bray-Curtis, but considering we're only looking at species richness, maybe Jacardian could be a better fit. We also want to explore alternative modeling structures that might help us solve some data transformation and singularity issues that we've been having. And finally, we're also interested in integrating a multi-trophic stability metric more explicitly. When we say that, we really mean right now we're doing everything with producer and consumer separately, but we're interested in potentially combining the data sets and assessing stability in that way. And in terms of publishing, we're planning to publish the harmonized data set to EDI, and we're ready very soon to start writing and submitting and eventually submitting the associated manuscript. And our group also submitted a Spark proposal, so hopefully we'll be able to continue this work either through that way or through other means. And with that, we'd like to say a huge thank you to Nick, Marty, Carrie, and everybody at the LNO and NCs for making our experience there really amazing. Thanks also, of course, to our mentors, Laura and Forrest. And of course, all of our Seeker program class attendees, everyone helped give feedback on these ideas, and we're grateful to them. And of course, this would not be possible without LTER network and the information managers. And with that, we'd be happy to take any questions.
Thank you so much, Nick. You should just be able to.
Yeah, but I feel like the vibe was. But I've asked you guys this on the side, and I think you had a good answer, but I'm going to ask for the benefit of everyone else. It looked to me like a lot of your sites have interesting relationships between stability and richness that aren't reflected in aggregate before you standardize. Have you considered testing a site interaction term instead of putting site as a random effect?
That is one of the alternative model structures we are considering.
Cool. Other questions for this group? I was really pleased to see that you're going to be looking for more terrestrial data. It seems like it must be out there. I think
Yeah, it was kind of surprising that we had so few. I think
Yeah.
we're being pretty strict about what we include or don't include. It has to be at least 10 years, and it
Right,
has to
10
be
years.
spatiotemporally co-located, and it has to be taken from within the site at the same time, basically. So if you have plots that are kind of scattershot, so where it's like 10 years over here and 10 years over here and 10 years over here and 10 years over here, if they're not overlapping for at
Right.
least 10 years, that makes it kind of hard to draw comparisons, especially given varying rates of disturbances within those systems that they may or may not encompass. But
Yeah.
We are going to look at the neon data set. I think there we should be able to find more terrestrial and aquatic sites that fit for our purpose.
Yeah, the 10-year limit might be challenging there. The earliest sites are just starting to get there.
Yeah, we talked with some neon folks, and we think there's maybe like one data set or maybe two that, especially if we like dropped it down to 9 years. But I think the time plus the trophic levels makes it kind of hard.
Yeah. Yep. Byron has a question. I think you can just.
Yeah. Okay. I'll just ask you about. I was wondering about the super awesome work, by the way. That's just really cool watching this. It's super amazing stuff. I wondered if you might predict that the shape or the direction of the relationships that you're looking at might differ in a predictable way between high diversity systems and low diversity systems, like tropical systems and polar systems or temperate systems.
I don't know if the shape would differ necessarily. I think that in general, one of our shortcomings is that we're reviewing these in terms of general linear relationships. We wanted to get more polar and high latitude data sets in general. They almost entirely did not meet our requirements, I think. But generally speaking, I'm not sure if we would see weaker relationships or not. I actually don't know. I don't know if anyone else has an idea here.
Go ahead, Junna.
I can try to answer this question. What I have noticed from our data sets is terrestrial sites has much less terrestrial sites is much less diverse than aquatic sites. So our aquatic ecosystem is highly diverse. Like we have hundreds of fish species, like all macroinvertebrate species in marine ecosystem. So in these systems, we see very strong positive relationship between diversity and stability. conversely, in the sites that we don't have a lot of species, like the one is salt marsh, the other one is the relationship is not necessarily positive and it's relatively weak. I'm not very sure if this is true or if we look at more study sites. But right now, emerging pattern seems more diverse system is likely the relationship is more positive. Okay.
Thank you.
Want to add?
And Mac asks, were the response and predictor variables scaled in the same way? Sorry if I missed that.
Yep.
Okay. Other questions for the group? And I will remind folks that all of the contact information for these, both the individuals and the groups, is available on the ssecr website. Carrie.
Yeah. First, just kudos on the science communication, y'all. Really nice job making a bunch of different comparisons and some complex methods accessible. I am intrigued by the idea of the multi-trophic stability measure that you were thinking about maybe exploring, and just wondered if anybody could talk a little bit more about that, if there are examples of researchers applying that in single site situations that you were inspired by, or what you think you might find with that.
Yeah, I mean, unless someone else wants to take it, I could take that too. But basically, the idea came from a paper that Griff Shrednick and Steve Sawyer published last year, where they basically and also work that Mary Donovan did back in 2022, I think, where they are explicitly combining basically animal and benthic or animal and plant data sets into a single species matrix, and either combining that in terms of their aggregate abundances or in terms of their multivariate species matrix. Where it got tricky for us was we were technically able to do that first with aggregate abundances, but when a lot of the data is percent cover, that means that the producers are necessarily going to be contributing less to that sort of multi-trophic abundance matrix. And then that also became an issue again when you start thinking about it in terms of the multivariate trait space, where the biomass metrics are just going to be dominating everything, and it wasn't really differing in the ways we were hoping for. I don't know, James, if you wanted to add more for the multivariate.
Yeah, just that it's going to be driven by a lot of the large biomass in the data set. And if we don't have good estimates of producer biomass in a lot of aquatic systems, it limits our ability to really get at that with those combined species matrices. I will say that like a lot of the data sets are pretty zero inflated, so they are being driven by relatively few or common species at a lot of sites, but how those trends would change or not, we're not entirely sure.